Today, we're going to talk about a piece of technology that is, and I'm not exaggerating here, like a flight recorder for a single living cell. It's called TimeVault, and it is completely changing how we think about biology.
So, let's start with a question. What if a cell could actually remember its past? Think about it like a detective arriving at a crime scene. You see what happened, but you have no idea how it happened. You miss the whole story. Well, that's been the problem for biologists for decades. So, what if the cell itself could tell you its own history?
And that gets us right to the heart of the problem, a cell's lost memories. See, our best methods, things like RNA sequencing, they're basically like taking a single photograph. It's a snapshot, frozen in time. You can see what the cell is doing right now, but you lose everything that came before it. The whole story is gone. Okay, to really get this, we've got to talk about something called the transcriptome. It's a bit of a sciencey word, but the idea is simple. Think of it as the cell's real-time blueprints. It's the full set of active messenger RNA, or mRNA, which are basically the instructions telling the cell what to do from moment to moment. And this right here really lays it all out. On the left, that's what we've had for years. A single static snapshot, a picture. But on the right, that's the holy grail. That's what we've been missing, the movie, the whole sequence of events. And filling that gap is exactly what TimeVault was designed to do. So how in the world do you build a time capsule for something as small as a cell? Well, it turns out the genius answer wasn't to invent something totally new from scratch. Nope. It was to repurpose a tiny little particle that's already hanging out inside our own cells. So, this is where it all starts. Nature basically handed the scientists the perfect container. It's this amazing little particle called a vault. And you can see why they called it that, right? It's literally a hollow, barrel-shaped particle that's perfect for storing things. It's a ready-made time capsule. Okay, so here's the really clever part. A hollow container is one thing, but how do you get the cell's messages inside? Well, the researchers engineered it. They added what you can think of as a grabber, a special protein that latches onto the tails of those mRNA messages. This little grabber just snags the cell's active blueprint and pulls it inside as the vault is being built. It's brilliant. And the whole process is just so elegant. It works in 4 simple steps. First, the cell makes the protein shell of the vault. Second, it also makes that mRNA grabber we just talked about. Third, the grabber does its job. It just reaches out and snatches up all the nearby messages. And finally, the vault just snaps shut, assembling itself around the cargo, sealing that moment's transcriptome safely inside. Okay, the idea is beautiful. The theory is elegant. But does it actually work? And maybe just as important, does stuffing a cell with these tiny recorders mess it up? The researchers had to put their new time machine through some serious stress tests. All right, this first test was a make or break moment. This chart shows how well the system captures mRNA. And look at this. The first 2 bars on the left, that's when you have just the vault shell, or just the grabber. You get basically nothing. It doesn't work. But then look at that 3rd bar. When you put them together, boom, the capture rate just skyrockets. This proves it. You absolutely need both parts. And once a message is captured, is it actually safe? Oh yeah. RNA is usually super fragile stuff. It has a half-life of only about 17 hours in a cell. But once it's tucked away inside a time vault, that half-life jumps to over 132 hours. That is more than 7 times longer. It means a memory from days ago is still perfectly readable. Now, for the real acid test. Can this thing actually tell the difference between the past and the present? So the researchers designed this incredibly clever experiment. They turned the recorder on, they blasted the cells with a quick stressor, in this case heat shock. Then they turned the recorder off and let the cells completely recover for 2 days before they looked at them. And here it is, the payoff. This is just incredible. On the left, that's the recording from inside the time vault. It is lit up with the genetic signature of heat shock. No question about it. But now look to the right. This is the current state of those exact same cells. The heat shock signal? It's gone. The cells have recovered and moved on. This is the smoking gun. It proves TimeVault captured a true memory of the past. So, what do you do once you've built a working cellular time machine? Well, you go after one of the biggest, most deadly mysteries in medicine: cancer. Here's the problem. In some lung cancers, when you hit them with powerful targeted drugs, most of the tumor cells die off, just like they're supposed to. But a small group always survives.
They're these tough, stubborn persister cells. And for the longest time, we haven't known why. Are they just lucky? Or were they different from the very beginning? This is where TimeVault is the perfect tool for the job. The plan was so powerful in its simplicity. Step 1. Turn on TimeVault and record what all the cancer cells are doing before you give them any drugs. Step 2. Add the drug and let it kill most of the cells. Step 3. Find the survivors, crack open their time vaults, and read the memories of what they were like right at the start. And this completely flips the script on how we think about drug resistance. It suggests the key isn't what the cancer cell becomes after you hit it with a drug. The key is what it already was before the fight even started. TimeVault finally lets us see that hidden, pre-existing identity. And here is the stunning discovery. By reading those before messages, TimeVault found 449 genes that were already different in the cells that were destined to survive. This isn't a response to the drug. This is a predictive signature. This is the blueprint for survival, and these cells had it from day one. And we're not just talking about abstract data here. We're talking about specific genes, the actual culprits. Names like FN1, PI3, CXCL8. These are the molecular breadcrumbs, the hidden clues that mark a cell as a future persister, long before it ever even sees a chemotherapy drug. And this right here is the absolute kicker. Over half of these predictive signals completely vanish after the drug treatment is over. So if you only looked at the cells at the end of the experiment, which is what we've always done, you would have completely missed the most important clues. They were only visible in the past. So you can see how this breakthrough just rips open a whole new way of doing biology. We're not just taking snapshots anymore. We're recording history. So what does that mean for the future? Where do we go from here? Well, let's put this in context. There are other molecular recorders out there, things based on CRISPR, for example, and they're amazing, but they're biased. You have to tell them what to look for. TimeVault is different. It's totally unbiased. It records the entire transcriptome, the whole shebang. It lets you discover signals you didn't even know existed. It's a true discovery engine. And the potential here is just massive. We can finally start asking some of the biggest questions in biology. How does a stem cell actually decide what it's going to be? What is the exact molecular flicker that kicks off a disease? Why do some of our cells seem to age faster than others? By linking a cell's past to its future, we can finally start to get real answers. In the end, what we've been given is a new sense, the ability to see through time, but on a cellular level. And now that we can finally read a cell's history, it really leaves us with the biggest question of all. What other secrets are waiting in the past, ready to be uncovered?